Welcome, everyone. We have a great workshop here this afternoon entitled Immunotherapy Beyond GD2. We're talking beyond dinutuximab there. We still may go after the target. For the oncologist, the role of T cells, NK cells, PD-1, and the microenvironment. So we have 4 longer talks, 1 shorter talk at the end, and hopefully have some time for questions and discussion. So without further ado, I want to welcome John Maris from the Children's Hospital of Philadelphia, Rethinking Immunotherapy Approaches to High-Risk Neuroblastoma.
Thank you, Crystal, and thanks to you and Ira for the invitation to kick off this workshop. And really, what I hope to do today is just set the stage for the great talks that are going to come after this about thinking new about how we're going to approach immunotherapy of childhood neuroblastoma. So I've used this slide several times, and just really I want to highlight that when we're talking with pharma and others, immunotherapy for childhood cancers can't be a trickle down from adult malignancies. They're fundamentally different in the fact that childhood cancers are developmental malignancies, very different lineage-specific drivers, whereas adult cancers largely are environmental disorders. We all know in this room, and neuroblastoma was one of the first to prove this, that the overall somatic mutation burden, which we think is a biomarker for immune checkpoint inhibition, is low, at least at diagnosis. And we know that changes over time with chemoradiotherapy. But in general, immune checkpoint inhibition has been inactive in childhood cancers, whereas it's been strikingly active in certain adult malignancies. CARs have worked in childhood ALL. They've worked in some adult malignancies, but the success rate of synthetic immunotherapies is certainly higher in childhood cancers. And then finally, and what we'll focus on a bit today, is that the proteins, glycolipids, et cetera, that are in the membrane of childhood cancers, in particular neuroblastoma, are quite different than those in the majority of adult malignancies, and this is something that we want to focus on. So everyone knows these slides. Alice Yu's seminal study showing that GD2 immunotherapy, directed immunotherapy, improves event-free survival and maybe has a small impact on overall survival. And while these curves for CART19 in childhood ALL may look similar at first blush, they're really quite different when you look into the details. And the bottom line is that this is curative therapy for many children with highly refractory ALL, whereas I would say that GD2 is credentialed as an active agent, but whether it's is contributing to cures is something that I think we still don't know. And it clearly has some liabilities, and that's why this session is entitled, "This is important, it's FDA approved, we have it, but can we build on this, but can we also look at other approaches?" So Crystal and I had the real privilege of pulling together a North American group 5 years ago now to focus on using the power of genomics to try to come up with new ways to discover immunotherapeutic targets and credential them in childhood cancers. And while this is a broadly pediatric cancer-specific project, I'm going to highlight today some of the advances that we've made in neuroblastoma. And this was from a review that Kim Stegmeyer wrote, and really our approach in many respects is pretty simple. We wanted to take largely RNA sequencing data, look for differences between tumor and normal, things that we can target on the cell surface, and then there's a bunch of different ways that we can go after this. But what I think made this simple approach a little bit more rigorous is that we were really looking for differentially expressed proteins, and we continue to look for differentially expressed proteins or other molecules on the cell surface that are critical to the lineage specificity of the cancer of interest and are essential for the sustenance of the tumor, and I'm going to show you an example of that. So when we started this project, there was extant knowledge of a variety of different immunotherapeutic targets across childhood cancers and a variety of different types of molecules, and we purposely wanted to stay away from MHC-presented peptides because of all the problems inherent with that, and I'll come back to that at the end, that we're now approaching that. And to summarize the advancements we've made over the last 4 years, in red are proteins that have been credentialed by one of the 10 laboratories that's been involved in this project as a potential immunotherapeutic target in a histotype, or more commonly in some sort of genomically defined subset of childhood cancers. And there are a lot of these potential targets in neuroblastoma. So circled here are all things that we are going to, are being pursued, some of them old, some of them new, GD2 obviously, L1 CAM obviously, B7H3, but many, many others that we think bear all the hallmarks of a cell surface protein that we can go after, or cell surface molecule that we should be able to go after with either antibody or cellular-based therapies. And I'll end by talking about a little bit of work that will be presented later in this meeting on MHC-restricted peptides that are presented. I'm going to start by talking about GPC2, and I'll speak a little bit about DLK1 as we go through this talk here today. So this is Chris Bossi, and he's giving a plenary talk on Saturday about this molecule GPC2. So here my goal is just really to talk about his published data, and I want to put it in the context of why we think this is perhaps a model of how one can use RNA sequencing data to define optimal immunotherapeutic targets. So a lot of work went into this figure. Chris went through, when we started the St. Baldrick's Stand Up to Cancer project, one thing we had a lot of was genomics data. So, and I apologize for those of you on this side of the room. We don't have a mouse up here, so I can only point to one. But this is what we were looking for. We were looking for genes that were expressed at very high levels, and so this FPKM, or measurement of RNA sequencing, of about median of 60 with some outliers, very high, is actually very high RNA expression. And then you look across, you can't read this, I'm sure, but a bunch of normal tissues. And there's a little bit in testes, a little bit in some other tissue, but really very, very restricted. And Chris has actually went ahead and shown, and I'm not showing that here, that there's a tumor-specific isoform in the normal tissues that's a different isoform. So this looks pretty clean, and this is what we were looking for in terms of immunotherapeutic targets. But what Chris did that I think is really important was it's not just differential expression that makes things a good immunotherapeutic target. There are many factors, but the two that we really honed in on is whether or not it was expressed because it's an essential component, or it plays an essential role in the sustenance of the tumor cell, so so-called oncogenic dependency. And Chris showed that very clearly in his paper that was published earlier this year, that when he knocked down, here using short hairpins, also with CRISPR, if you knock down the protein, you get very, very significant decreased proliferation, here using cell titer CLO, decreased proliferation in a real-time cell monitoring assay, decreased clonogenicity. So these cells just do not tolerate depletion of GPC2. And also, very importantly, that this induces programmed cell death here with cleaved caspase and caspase assays. So Chris showed this in multiple different ways, that the cells don't just stop dying, just don't stop growing, but they actually die. The other very important thing that we want to try to understand as we nominate other molecules for potential immunotherapeutic targets is what is the mechanism of overexpression. And I think Chris very beautifully first noticed the correlation that there was higher GPC2 expression if in tumors that harbored MYC amplification. The gene is located on 7q, and there was also increased GPC2 in tumors that had 7q gain. And if you had both of those, you had the highest expression. And that was interesting, but what Chris then went on to do was to show that GPC2 is a classic MYC target gene. There's an E-box, and using ChIP-seq in several of our cell lines, you can see that MICN is binding right before the GPC2 promoter, and if this is if you overexpress MICN, it drives GPC2 expression, and that if you deplete MICN, you decrease GPC2 expression. So we think that the fact that this is a MICN target, there's a genomic, the MICN amplification and 7q gain are almost mutually exclusive, so you have one or the other. Both mechanisms drive GPC2, make it a very good target. And then Chris went ahead in this paper to show that if you build an antibody-drug conjugate, these are things that we now can do rather rapidly in academia to credential these targets. This is a PBD-containing humanized antibody against GPC2 that you can, a single dose can regress tumors in mice and cure these mice over many, many weeks. And Chris will be expanding upon this both to other pediatric cancers, small cell lung cancer, as well as showing a lot of other interesting data about the efficacy of this molecule in his plenary talk on Saturday. So GPC2 is an example of the RNA expression, thankfully, very much predicts what the protein expression is. But I think we all know that that's probably true only about half the time. And there are nice published data from all of the proteome profiling projects that, there is, if you just look at, I'm sorry this got cut off here, this is protein expression by mass spectrometry, this is RNA expression by RNA sequencing. There are some molecules where there is a good correlation, but there are some where there's absolutely an anti-correlation. And so as we moved into the later years of our St. Baldrick Stand Up to Cancer project, we really wanted to bring in proteomic approaches to help us verify predictions from the RNA, but also perhaps identify unique immunotherapeutic targets in neuroblastoma and other childhood cancers. And so this is a complicated slide from Paul Sorensen. There's really three main ways that people go about taking a high-risk neuroblastoma, PDX or primary tumor, and trying to figure out what is the proteome of the plasma membrane. One is just to do whole cell lysates and then try to figure out what was plasma membrane. We don't like this one. There are both labeling methods and label-free methods. And Amber Weiner, as I'll get into in a minute, has really mastered this sucrose gradient purification of the plasma membrane. And that's what we've gone forward with in a way to try to get an enriched resource of plasma membrane proteins to put on the mass spectrometer and get an inference of what are the proteins that we might be interested in. And so this is Amber, and she'll be presenting a talk on Saturday in one of the parallel sessions that will expand upon the very little tidbit that I'm going to give you here today about the work that she's doing in Sharon Diskin's lab, as well as in my lab. And this is what her mass spec data, this is just from the cell lines, so she spent a lot of time using human neuroblastoma cell lines to sort of, to get this highly reproducible, and she'll show some of those reproducibility data on Saturday. But this is this swoosh plot of there are a lot of proteins that are on the low end, a lot of proteins that are at the high end, and what was reassuring to see that a lot of the things that we know about are up on the higher part of the curve. And as she'll expand upon on Saturday, there are things that we had heard of but really hadn't focused on just from the RNA sequencing, such as DLL3, and I think many people know that this is something that AbbVie is going after with a antibody-drug conjugate, and Renata Sano and Yael Mosse's lab presented on this at AACR, as well as this molecule DLK1, where there's a little bit of literature of it in neuroblastoma, but this was a bit of a surprise to us. So as Amber will get into on Saturday, if you just look at the gene expression, unlike GPC2, where if you looked at a bunch of high-risk neuroblastoma cases, I think this is the German data, RNA sequencing data, you would see almost all of the cases show all the high-risk cases show high expression. You really have this, there's a bunch of low expressors and a bunch of high expressors, and sometimes it falls out that it's MICN amplification that is correlated with the expression, and that was not the case here. So what Amber cleverly figured out with Jolyn Horenza and Sharon Diskin is that it is likely that this protein is driven by the acquisition of a super enhancer during tumorigenesis or tumor evolution. And so this is a look at DLK1 expression across our neuroblastoma cell line panel. And we have performed H3K27 acetyl and a bunch of other chromatin marking studies in these lines. And they segregated it into two categories. There were several of these, and I think many of you are familiar with these rankings, another way to look at another type of swoosh plot where you're looking at the rank of the genes that have the highest enhancer mark or super enhancer mark with many of the things like FOX2B and MYC-N way up here. And you can see the DLK1 scored very highly in most of these. GPC2 is down low because it doesn't need a super enhancer since MYC-N or 7q is driving it. And then there are others that there was DLK1 is way down at the bottom, and it teed up almost perfectly that those with the super enhancer mark were the high expressors, those without the mark were the low expressors. And again, this gives us a lot of biological fortitude of why we might want to go after this, and we are collaborating with a company that's made an antibody-drug conjugate against this molecule for testing in our preclinical model systems. And just very briefly, as part of a new project, and Paul Sorensen is driving this, we're going to be doing a lot of this, basically a pediatric cancer plasma membrane proteome landscape in a lot of different tissues. And this is Amber's analytic pipeline that she'll talk more about on Saturday. So I want to end with, again, setting up another talk that will happen during this session by Mark Yermarkovich. And he came to me as a very talented graduate student who came to me and was not worried about the well-known problem that I already mentioned, that neuroblastomas have a low mutation burden. Old literature that they are very famous for having low class 1 MHC expression, and that they are the quintessential cold tumors. When adult cancers want a model of a cold tumor, it's generally neuroblastoma they pick, because what this is showing is there's no T cells in the majority of these tumors. Now, we know that some of the MYC-N not amplified, there might be a little bit more of a T cell infiltrate, and that will change with chemotherapy, but they're generally the quintessential cold tumor. So, again, Mark will be talking about this tomorrow in one of the parallel sessions. And Mark developed this really elegant pipeline, and I'm going to go through it very briefly because he's going to do it again tomorrow, but it really is based upon a MHC-based pull-down of and mass spectrometry to elute peptides that are bound to MHC specifically, to do this in a large number of neuroblastomas and compare it to a very large number, 190 different healthy tissues where this so-called ligandomics has been performed. All of these tissues also have DNA and RNA sequencing, and this gets integrated to try to understand which peptides are differentially expressed in not on normal tissues. And Mark will go through this in a lot more detail tomorrow. And this is what the data look like. And again, I don't want to steal much of Mark's thunder, but what this is plotting out is this is peptides eluted from the neuroblastoma tissues, and these are peptides from the neuroblastomas. Do I have that right, Mark, or am I backwards? Normal on the bottom. Okay, thank you. So things like this, where there's a lot of peptide in neuroblastoma, but there's peptide in normal tissues we're not interested in. And what Mark honed in on were these peptides that were uniquely expressed in neuroblastoma. And it was really cool to me when he came to the office and said, when I take these peptides, map them back to their genes, and do a go analysis, the only thing that comes out is sympathetic nervous system developmental proteins. And so that gave us a lot of interest. So the very, very other important thing is that none of these were neoantigens. These were all self-antigens. He didn't find a single neoantigen in this experiment to date. And Mark will present tomorrow that through a lot of prioritization of these developmental, many of you will recognize FOX2B, TH, and what he has shown is that there are many of these peptides that he's been able to elute that come from proteins transcription factors generally, lineage-specific transcription factors that are highly expressed in neuroblastomas and absent in normal tissues. And we think that these are good TCR targets, and Mark will present some early data of his cloning of T-cell receptors and how he's shown early proof of efficacy of this approach in neuroblastoma. So I hope I showed you today that GD2 has clearly been credentialed and is an important, very important first step. Waiting for Godot was a long time coming, but there's been a lot of progress. But it has some liabilities, and we think we should look beyond GD2, which is the title of this session. We think that there is a lot of promise in many of the other molecules that are present on the cell surface, and many of our laboratories are working on development together of antibody-drug conjugates and CAR-based approaches against these molecules in collaboration with making biomarker or understanding biomarkers. And we're really focusing on single-cell sequencing and other ways to look at cell-to-cell heterogeneity and the likely necessity of bivalent approaches. And I think Mark has shown that going after the MHC-restricted peptides in this disease is possible, and that's something we're very excited to pursue. So I'd like to thank all the members of my lab, many of whom are here, Yael, Sharon, Kate from adjacent labs, Crystal, I'm sorry, I dissed you. Crystal and Paul Sondel, everyone in our collaborative North American project, and other collaborators. So thank you for your attention.
That's great. We have time for a couple questions.
So we biased our... So the question is you know, I made the comment we didn't find any neoantigens, and I should have made it very clear, and thank you for the question, that we're not saying that there aren't neoantigens there. We biased this experiment to have tumors that went in that had mutations, ALK mutations, other mutations, and were predicted to present, you know, all the prediction algorithms said neoantigens should have been there, but we didn't find them. We're probably only getting 10 or 15% of the peptides in this experiment. And so I don't know if they're just at a lower level and we haven't seen them. Through a completely different collaboration, we have found ALK neoantigens using a different approach. So I'm sure they're there. But what we really are excited about here is the recurrency of these. And that's another point that I didn't emphasize, is that many of these self-antigens are recurrent on common HLA alleles, which is why we think we might be able to leverage that a little bit more than the neoantigens.